This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 340, recorded on June 5th, 2015. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast all about viruses. Joining me today from Singapore, Dickson Despommier.
Hello, Vincent.
You just got back, huh?
Yeah, but just about an hour ago.
I'm so sorry.
I'm not. I'm glad to be back.
Happy birthday.
Thank you.
You're welcome. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Is it your birthday?
It is not.
All right.
But it's 78 degrees, partly cloudy. Pretty nice day out.
It's all cloudy here.
It's raining outside right now.
17°C here, yeah. It's been rainy and cloudy all week, Dickson.
Yeah.
How is the weather in Singapore?
96 to 98 degrees and 80% humidity. Although, how would I know? Because I spent the whole time indoors.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here. It's much nicer here than it is there, apparently.
Really?
Interesting.
Yeah, we have. It's a little cloudy. Few clouds, but they're fairly high up, and I guess there's an overcast layer much further up, around 13,000. But yeah, it's 74 Fahrenheit, 23 C, no rain. Cool. Lovely day. Light wind just coming in out of the south.
Excellent.
Now let's get the real weather. Also joining us from North Central Florida, Rich Condit.
Hi, everybody.
Hey, Rich.
We got 88 Fahrenheit. That's 31 C. We got, I would say, partly cloudy or mostly sunny or something like that.
Yeah.
They're nice, white, puffy clouds. It's not obnoxious. Humidity 51%. People are going to want to know that. The dew point is 68 degrees. So that's, it's a little, if you were from San Diego, you'd think you'd probably die, okay? But for us, it's okay.
What's the magnetic flux?
I don't have the magnetic flux here, sorry.
Are we up to another reading? How many?
Just a minute here. Let's see. Solar flux is 119. That's off 80 sunspots.
Wow. Jeez.
So if that's what you wanted. The A index is 3, the K index is 1. So actually pretty good conditions for the ionosphere.
Pollen counts, anybody?
I don't have pollen counts handy.
Rich Condit, I thought you were going to be out this week.
No, I was out last week. This week I'm cool.
Because you weren't on the schedule. So I left you off. And
Oh, do you want me? Should I go away?
No, it's okay.
No, stick around.
I was just confused that Dickson was not supposed to be here either. He told
me.
It
could be that I was lax in keeping
okay.
up with the schedule.
No problem.
No, I'm all set to go. I'm ready. I'm ready to roll.
Yeah, I need to get back in the swing of this.
When's your next trip, Dickson?
End of June.
Good. Very good.
I thought you were going to say, when were you in the swing of it?
Never.
All right, we have some sad news to start. Nice. One of our virology colleagues has died, Richard Elliott, from Glasgow. He was on TWiV 177, was a Bunya virologist, and I knew him. He was a postdoc in Peter Palesi's lab when I was a student, so I knew him quite well. And I am going to Glasgow on Sunday, and he won't be there. He was supposed to do a TWiV with me.
It was a sudden incident?
Apparently, he went into the hospital 3 weeks ago. He's been sick for a while, though, and just died. And he had actually moved. I think I got him on TWiV 3 months ago. He said, he would do it, and then a few weeks ago, he said he couldn't. So it's too bad. I'm sorry. He's a nice guy.
Yeah.
And a good scientist. So we'll miss
That's
him.
a loss.
Yep. It's too bad. We have a follow-up from last time. Trudy writes, Dear TWivers, in answer to your question about who else out there has listened to all of the episodes, the answer is me. I've listened to all episodes of TWiV, TWIM, and TWIP. As a result, I'm very familiar with Twix culture. I also tried urban agriculture, but it's not my cup of tea. I like infectious disease. Keep it up, Trudy. So we had someone last week who listened to every episode.
That's amazing.
Wow.
Pretty cool.
Yeah.
I've listened
there should be a support group or I made up fan club. there.
We have a follow-up from Rich Condit.
Yeah, so I've been listening to TWiV in my absence. First was I want to make sure that this is a dying horse, I know, but I can't let it go. There's been conversation about the relationship between mutations in IRF7 and cancer, right?
Right.
So, and that came up because of some sort of reference in the paper to that.
I'm not sure because we wrote to Casanova and he said he never brought it up. So maybe it may have come up. It may have come up in a discussion subsequently. The general idea that cancers have defective innate immunity and that IRF7 is obviously a defect. So that got thrown in there, I think.
So Trudy, how did we end up on this subject?
Yeah, right.
So, but we did. As part of that, there was a discussion of the overlap between innate immunity and control of the cell cycle and that kind of stuff. Is that correct? Because that's what I wanted to. That's what I wanted to bring up. I'm reminded of the TWiV from Pat Moore, TWiV 160, that I wound up incorporating into my lectures because he made some very important points there about the degree of overlap between what we think of as antiviral pathways on the one hand and cell cycle regulatory pathways on the other hand, and how lots of times if you mess up, if a virus messes up an antiviral pathway, a consequence of that, since there's overlap between that and growth control, is to transform the cells. All right, and so I have incorporated into my regular thinking the notion that there is overlap between antiviral pathways and pathways associated with growth control. So the notion that a gene that we ordinarily think of as as involved in an antiviral pathway impacting on oncogenesis is, let's say, familiar to me. And I would refer people back to that Pat Moore episode because it's really good.
All right.
Cool.
All right. Herpes trashing termination. Yes, I'm sorry I wasn't present for that. So I think my biggest comment on this is that throughout this, they talk about termination. And I want to reemphasize that polyadenylation and termination are two distinct things, and people tend to conflate them.
Right.
And if we're going to look at this ultimately down the road mechanistically, it's important to understand the difference. So the polymerase travels down the DNA. It goes through a polyadenylation signal that recruits to the polymerase all of the stuff necessary for cleavage of the messenger RNA and polyadenylation. And most people at that point say it's done and forget about it. And lots of people, I think of that as termination. But that is not termination. The polymerase keeps going. The polymerase does not necessarily disengage from the template at that point. All right? Termination is when you actually release the polymerase from the DNA and release what's called the ternary complex, DNA, RNA, polymerase. But after polyadenylation, the polymerase keeps going, keeps making RNA, but that RNA at the 5' end doesn't have a cap or anything, so it's very labile. And Kathy, when we were talking about this before, brought up this torpedo nuclease that's a 5' to 3' exonuclease that follows this thing along and is trimming the RNA all the time and is implicated in the actual termination reaction, which is somewhere downstream from the poly-A site and can be way downstream from the poly-A site and can happen at multiple different sites, at which point the polymerase disengages from the DNA. But that RNA that it's making at that point is so labile that nobody ever sees it, nobody ever thinks about it. So I'm thinking in this herpes paper, they don't really distinguish between, all they know is the RNAs get longer, even to the point of reading through downstream genes. And they don't distinguish between polyadenylation and actual termination. And in fact, there is a relationship because the polyadenylation reaction does impact. I think it stimulates termination. So if you don't have polyadenylation, you might read further than ordinarily. But mechanistically, if I were looking into this, I would take a close look at herpes tinkering with the poly-A machinery. Okay? And I don't know, in the case of herpes, whether the polyadenylation and termination, I don't know how well that's understood. I would assume that it uses the host apparatus for this because it's using host polymerase, but I don't know if it uses a modified version of it at all. And that's worth looking into for understanding how herpes manages to, in a herpes infection, this is specific for the cells and doesn't apparently impact the virus. The other thing that struck me about that paper was that I believe the comment was made that it seemed striking that RNAs that were read into genes from upstream were not translated. And to me, that's not striking at all, okay? Because in order to get translated, you have to start in the right place and get capped right so that the cap that attracts the translation machinery is properly positioned relative to the translation initiation site. And in fact, this goes the same for splicing. Splicing is a co-transcriptional event. And if you don't start in the right place and configure everything properly, you're going to mess up splicing as well. So these RNAs that are read through from upstream genes and stuff, they're garbage. All right, and it doesn't surprise me a bit that they're not translated. Doesn't surprise me at all that they're not spliced properly or anything else. That doesn't dilute anything about the paper. It just gives these are thoughts that I had about it as it was going on. But I'd have a close look at the polyadenylation apparatus and see what was up there.
Yeah, I think that's a good idea. Yeah.
Rich, we're talking about the fact that the polymerase goes on and then the RNA that's made is relatively unstable. Do you think it's possible that somehow herpes is stabilizing that?
I suppose, but you would have the same effect just by compromising the polyadenylation. Okay, you'd continue to read on through. You know, I think you could do all of this by trashing some aspect of the polyadenylation, the cleavage, probably the cleavage complex, something like that. And this is all tied in. I don't know the details. It's also all tied in with the phosphorylation status of the C-terminal domain on the polymerase. That controls a lot of this. Blah, blah, blah. That's where I'd be looking.
All right.
Okay.
Thank you for your insight.
Sure.
I thought you would like that.
I was talking to you guys.
Yeah, I'm sure.
Yes.
Yeah, because that the termination, trashing of termination, or whatever you want to call it, because we don't know what's going on, happens late in vaccinia virus infected cells. Right?
Well, in vaccinia, it's the situation is different because both early and late during vaccinia infection, the vaccinia does not use any of the host machinery, and it doesn't use the host. Doesn't follow the host paradigm of a cleavage and polyadenylation. Okay, it's all just apparently just a termination and polyadenylation. Okay, so it skips the whole cleavage thing, and the termination reaction late during infection looks similar to what I envision real termination in eukaryotic cells to be. That is way downstream from the normal coding sequence and in multiple different places. And it's a real termination. The polymerase disengages from the DNA. So it's a different kind of story.
Do we know whether any of the other large nucleocytoplasmic DNA viruses do similar things? I guess we haven't studied any of the others.
Most of them contain a homologue to my favorite protein, A18, which is the termination factor that is used late during a poxvirus infection. Okay? So, and most of them also contain homologues to other proteins that are involved in that same reaction. So if I had to guess, I would guess that the processes are similar, but nobody's looked.
Okay.
Right? I was going to say one other thing, actually, that came to me. You mentioned, I think, Vincent, you mentioned northerns, okay? And this is an example of what a lot of people now think of as old timers, archaic technology that is so much more informative than a lot of this stuff, because all of this sequencing and PCR and all this stuff, you don't get to see what the structure of these things is. Yeah. And if you just did a northern on some of the host RNAs late during a herpes infection, matter of fact, if I were reviewing that paper, that's what I would have asked. I would be very interested to see a northern.
Yeah, of course.
In uninfected and infected cells, because there I would expect that what will happen is if you had something like GAPDH that ordinarily gives you a nice discrete transcript, that in a herpes infected cell you'd see this big smear.
Yeah,
right?
Right. Yeah,
and one simple experiment has the whole picture in it.
Yeah.
Northerns are cool.
That's actually why I was a bit late here. We were discussing one of my students' progress with her northern blots. She's complaining that we don't have the right equipment because we haven't done it.
Because nobody does them anymore.
Yeah. What equipment?
It doesn't take a lot,
right?
It doesn't take a lot.
Well,
you need a horizontal gel app and a pump.
There you go.
We have that, and she doesn't like the idea of stacking paper towels up. So I went online and showed her the recommended way to do a northern is to stack paper towels up.
Not only do you stack paper towels up, but you need to stick a glass plate on top, and then you have to have your favorite icon to go on that. Okay, one of my students had one of these figures of some superhero holding a big sledgehammer. So Batman would do. Okay.
Oh yeah, we have had the
Blotman. It's Blotman. Sorry.
The raisins.
Yeah.
And we've had Dino the Dino.
Yeah.
Somebody I know used to put a set of teeth. Her husband was in dental school.
Set of teeth.
Yeah.
All right, we have a couple of snippets, two of them dealing with MERS coronavirus, and South Korea back in May had its first case. It was an imported case in a 68-year-old male coming back for Bahrain.
Via Qatar.
And now we have had, 36 cases and 3 deaths.
4 and 40 is what they said on NPR this morning.
Wow.
What the hell does NPR know?
I think they check their sources.
Why is ProMed behind NPR?
I don't know.
This is from 6/4. This is from yesterday. All right. So all of these are preventable infections because this guy, they put him in a room with somebody else,
right?
And that person got sick, and then the healthcare workers got sick, their kids got sick.
Somebody went out and played golf, left the hospital early.
This is a lack of control. I guess they wouldn't know that he had it right away, but...
I gather it took some time for him to get diagnosed, which is not surprising.
Yeah.
Right.
And along the way, there was... I don't know if this has been confirmed, but there was speculation he might have been intubated.
Oh, my.
Jesus.
Which is not unusual for somebody who's having trouble breathing. This is a standard thing that you would do. But if you're dealing with a virus that is deep in the lungs and is not easily spread person to person because it tends to be deep in the lungs, and then you intubate somebody, you may aerosolize the virus into the room, and if you're not using the precautions that you would use if you knew this was MERS, then you get healthcare workers infected, and that's where this may have come from.
And many of these individuals are rather advanced in age. Not all, but I'd say a 45 is advanced in age. No, just kidding. 77, 60,
anything
50,
over 75.
59, 73, 78, 57,
but there are also
55,
some
58,
40,
77.
35,
Yeah.
58, yeah.
Is there a 30-something here?
35.
35s.
35
But you don't see any 20-somethings.
What's
No,
interesting is that they
20-somethings,
closed the school system in Korea to prevent spread of MERS, and you don't see one single person that age in this group.
Right.
All I heard on the news in Singapore was all about this outbreak in Korea, and it's killed the tourist industry, and it's it's a huge economic impact on that country.
Well, I think closing the school is not a bad idea because the teachers are going to get infected.
That's probably right. Yeah,
so
that's true.
Kids can.
That's true.
In fact, kids.
But they weren't doing that to protect the teachers. They were doing it because they.
But
didn't want
most of the
kids
people
to
so
go.
far are medical staff or people who were or other patients in the hospital.
Yeah.
So here, this 43-year-old had a history of pre-existing comorbidities. So maybe some of these 40-somethings and some of them. So that's unfortunate.
Yep.
So that's it. I just got an email this morning, actually, to talk about this a bit. Let's see if I can dig it up here so we can incorporate it into the show notes in a timely manner. Yes, this is from Lisa. Dear TWiV team, I teach English as a second language, and many of my students are living and working in South Korea.
Right.
These days they have become very distracted from their English studies over concern about the recent MERS outbreak. I have run out of things to say to them to assuage their fears, such as it is spread through direct contact, and it is very unlikely they will contract MERS if they are not healthcare workers. I have suggested taking vitamin C and extra hand washing, but most of my students are freaking out, and I feel that while their concerns are valid, they have reached peak panic mode. Lately, they have told me that they don't understand why their government will not release the names of the hospitals where MERS patients are being treated, and I've tried to explain that it is probably due to patient privacy, among other mitigating factors. South Koreans are very distrustful of their current government, and they are habitual users of social media where it seems rumors are spreading faster than the virus itself. They have blamed their government for the fact that their patient Zero went from hospital to hospital before anyone diagnosed him with MERS. Can you please do a show about MERS or speak about the South Korean outbreak during a portion of one of your upcoming episodes? I would really like to hear your take on this outbreak, and hopefully you can give me some information about MERS that will help me to talk my students off the edge. I majored in English literature and psychology, so this is not exactly my area of expertise, though I do enjoy virology and your show. It's a sunny 65F today in Cincinnati, Ohio, with an expected high this afternoon of 84. We have 87% humidity, winds 2 miles per hour. Lisa.
So I wonder, how long has this been going on in South Korea?
May 20th, I think.
May 20th, so we're talking about 2 weeks at least?
Yep.
I wonder how many people in South Korea have died on the highway in the same period of time.
Well,
Yes.
no, that's true. And the other thing is that everybody's wearing a mask, and if it is direct contact, then obviously that's the wrong approach there too. But somebody who sells masks is making a lot of money.
Oh, sure. That's the standard thing in all these cases. Everybody starts putting on, rushing out to buy masks and then not wearing them correctly,
Right.
Which it's not protecting you from anything. But I'm not sure there's anything to say to talk people out of a panic over this if they've already made up their minds to panic. But the government's actions don't look in any way suspicious to me. I don't know if that helps, but of course you wouldn't say the names of the hospitals. There's the patient confidentiality issue. There's also the issue of healthcare infrastructure, because if you name those hospitals, everybody will panic and flee from those hospitals.
Right.
And then
got it right.
adjacent hospitals will be forced to pick up the slack, which means that if there was anybody there who had contracted MERS, for example, by sharing a ward with one of these patients before they were properly diagnosed, they're now going to move to a different hospital, restarting the same problem. So no, you wouldn't release that information. I think they've released reasonable amounts of information under the circumstances. This is not like China with the original SARS outbreak, where nothing was said for a long time and things just kind of festered. This is a situation where it seems like it's being handled reasonably well now, although I might have some quibbles with the way it was handled initially.
But I think closing the schools sent a mixed message, because it indicated that perhaps it can spread by droplet infection, which it can't. But so people took precautions along those lines, and that's the wrong approach, obviously.
So the vast majority of these cases are in people who are in direct contact with somebody who is sick in the hospital.
Right.
Right?
Yeah.
I think there have been a couple of cases where somebody was in contact with somebody who was in contact with somebody in the hospital, and got that secondary contact eventually got sick. But I think there were some tertiary cases, if correct me if I'm wrong, where somebody got it from somebody that before they were sick enough to be hospitalized.
The transmission is from sick people, as far as we know.
All from sick people.
Yeah, in fact, in the next story, we're going to see that mentioned directly.
And so,
But they do say that there are 2 cases that were tertiary cases.
Right. But so
well, right, they're tertiary cases. They got them from people who got them from
exactly
people in the hospital, but then those people got sick, and then the tertiary infection spread.
So if you are not dealing with anybody who is sick in the hospital, you're not going to get this disease.
Right. So why close the schools?
And the hospitals are now very, very aware of this.
Yes.
So if you show up with a cough at a South Korean emergency room, I'm pretty sure you're going to be isolated pretty quickly. But yes, the closing of the schools, I don't know why that was done, but if I had to guess, I would say because it was something easy for the government to do to make it look like they're doing something.
Well, the problem with that is it makes a connection between SARS and MERS. And
it does.
Obviously, there isn't, but the Koreans certainly remember what it was like when SARS was around. So I listened to a lot of banter and rhetoric. And by the way, the president of Korea is a woman, and she was the one that issued that edict to close the schools. And she had to defend herself on television, and she was very inept at doing so. She circumvented all of the questions.
That's not good.
No, not.
Yeah, that's what politicians do, right?
Indeed.
To me, I'm just reminded about when, there were 1 or 2 patients in the United States with Ebola, and there was this semi-widespread panic. And
that's right.
I would like to say, and how did that work out for you?
Yeah, right.
To, all those people who were panicked.
Exactly. Well, ignorance breeds more ignorance.
Sure, but I mean, it's the same thing. When Rich was saying it, if you're not in direct contact with people who are sick in the hospital, there's probably not that much to worry about.
And at that time, there was something circulating on Twitter where someone pointed out the fatality rate on the highways and a survey that indicated the amount of concern about Ebola, and then they said, so therefore several thousand people who are currently freaking out about Ebola are going to die in car wrecks.
Right.
Yes, that's right. Yeah, so that is misinformation, and that's too bad.
But also these students are probably in their 20s or less, right?
Oh, yeah.
That too.
So these are not what we're seeing in fact.
They're not high risk, and they're not,
Right.
She could encourage them to listen to TWiV.
You can encourage them to listen to TWiV.
Right. It would help their English. And
it's homeschooling. That would be homeschooling.
Yeah, we could put some links. We've done quite a bit on MERS previously, right?
Yeah.
So we could put links. Although now we have a little snippet coming up now on MERS as well, which is interesting. This is a paper that was just published in the Lancet Infection in May. Presence of Middle East Respiratory Syndrome coronavirus antibodies in Saudi Arabia, a nationwide cross-sectional serological study.
This is really interesting.
So this is the really
cool.
first big, well-done survey. And we've been talking about this on TWiV quite a bit. We need to know who has antibodies to this virus.
Right.
And we don't know the number of people who have had infections. One of the problems here is that we know that the virus is in camels, but most of the infections, and there have been, over 1,000 MERS infections in that area so far. Most of the people don't have reported contact with a camel. So where's the virus coming from? So in this study, they got serum from 10,009 individuals. They have people older than 15 years of age, all over Saudi Arabia, 13 provinces. They also got serum samples from shepherds and abattoir workers. And in the abattoirs, they abattoir camels. They kill camels for various reasons, so these would have exposure. And they looked for reactivity with virus by ELISA and immunofluorescence and plaque reduction neutralization tests. So 10,009 individuals, they found antibodies in 15 people. 15.
Isn't that's very telling.
It's very low.
0.15%.
Very, very, very telling.
I'm surprised, actually. I would have thought you would have had more than this.
I want to ask a question. Has anyone done follow-up on the survivors of MERS to see how long the antibodies last?
I don't think so.
Because that's important here, because if the antibodies are short-lived, then this survey is meaningless.
But they also point out that, this is a survey of 10,009 people. If you multiply out the population of Saudi Arabia, you'd expect something like 45,000, almost 45,000 people might be seropositive in
Yeah.
Saudi
Is
Arabia.
there no clustering in any of the provinces?
I think significantly higher in, well, here's the problem. Higher in central versus coastal provinces and higher in the slaughterhouse workers.
Camel exposed, yeah.
But this is only 15.
Right.
Yeah.
You can't say it's too little.
So they say 23 times higher in slaughterhouse workers. But,
No, that's
you know,
not a good
That's
rate.
5 of them.
Sorry.
yeah, it's no good. So the number, I was talking to Steve Morse about this the other day, Dickson, and he said, it's just too few.
And-
Yeah, and it was 15 times higher in shepherds. That's 2 people.
Need a bigger
Yeah.
N.
Yeah.
Well, 10,000 was a big study to begin
That's
with, right?
yeah,
Yeah.
how many people are going to stick? But it does show that
But blood
people are
banks are-
probably getting exposed to this and not developing symptoms. That the population infected is probably larger than the population we're seeing.
Yeah.
Yeah.
So they actually say in their discussion the true number of infections is really hard to estimate because
Right.
we only have 0.03% of the population, which is 30 million people.
Does this also mention the case fatality rate in passing? Just wanted to refresh my own thought about that.
The case fatality rate, yeah, they have the numbers in the beginning, right? It's
Right in the introduction, yeah. 439 fatalities since its discovery in 2012 out of at least 1,082 severe cases of respiratory infection.
So that's a pretty high
about
case
40%,
fatality rate.
right?
That's pretty high.
yeah. But 15 is not going to make much of an impact on that.
No.
I mean, these are presumably 15 people who never reported any illness and of any severity to make them go to a hospital, right?
Right.
Right. They used sampling through a study that they already had of healthcare clinics for the national burden of disease was one of them, and a national study of seroprevalence of herpes simplex virus.
So they say here that they have they have people 15 to 44 years old who are seropositive in this study. So they're wondering if, these in this age group you get subclinical infections, and then
15.
those and they infect other people who have more comorbidities or whatever.
Could these be carriers? Yeah.
Yeah. But so that's interesting and makes a lot of sense, right?
Yeah.
But I think it would be nice to have more numbers. Although
Absolutely.
it's, what are you going to do? 100,000 people?
Right. It's,
I mean, you could do this in schools, for instance, and in institutions where you have a lot of people.
Well, this sample was people older than 15 years.
Yeah, right.
Because you could do like a filter paper, a drop of blood, and then just ship them to a central facility for analysis. They do a
Anyway.
lot of that in.
So I think this is
Yep.
a nice study. It was very interesting.
It's from a large collection of researchers from Germany, including Bonn, Cologne, and Lübeck, and Kingdom of Saudi Arabia, a couple places, Utrecht, and somebody from Indianapolis, IU School of Medicine. Oh, and Edinburgh, too.
Very good. All right, our last snippet is a story that came out a week ago today in USA Today, and it's called Biolabs in Your Backyard. And it is a real... Unfortunately, when you go to the link I'm going to put in the show notes, I think that's Mark Lipsitch there in the front, right?
yes.
Talking. A talking head. He doesn't make any sound. Well, I got my sound shut off. This is a USA Today network investigation. Identifies more than 200 BSL 3 and 4 labs that work with dangerous pathogens and reveals safety records that some of them fought to keep secret. So what they did, I know this because I'm on the Institutional Biohazard Committee here at Columbia, and every time we have a meeting, which is once a month, we have notes, we have minutes, right? And they filed a Freedom of Information Act for our minutes, and they presumably did so at all of these 200 laboratories across the country. And so we redacted some of the information, but you can go to your state. And so this is really sleazy. You click on the View Your State button, and then it says click on, so let's say we click on New York right here, all right, and then it says, here are the facilities that have BSL 3 or 4 labs. So they have Ian Lipkin's lab at Columbia because that's the only BSL 3 here. And then if you click on it, it tells you download the safety and accident records, okay? Those are the PDFs that they got, which are basically the minutes of the Biohazard Committee meetings. They are not safety and accident records only. They are all kinds of stuff. But, they want you to think that this is all that happens in these places, that they're accidents. Of course, in our notes, and if you go and look at the notes for New York City, you will get the IBC for Columbia, and you'll see my name is on every one because I'm on the committee, and we talk about all sorts of stuff. Sometimes there are incidents, maybe a needle stick or something spills or whatever. We talk about what was done to mitigate it. I don't understand why this is a big deal.
As a matter of fact, this is evidence that these people are monitoring their activities and keeping on top of it.
It works, right?
Yeah.
And, they have Lipsitch saying this is really sad to see all these incidents going on. He's amplifying his usual story, which is absurd. I mean, I don't see the story in this. Alan, any thoughts?
This was inevitable. So we've had a lot of isolated news stories about different issues in biosafety, right? We've had the stuff at the CDC. There were the smallpox vials, which, we've talked about all these things on TWiV. And then meanwhile, in the background, there's this drumbeat of Lipsitch and colleagues trying to ban research that personally scares them for some reason I can't comprehend.
Don't forget the live anthrax that just got uncovered also.
Right. And of course, a lot of this stuff is completely unrelated. Lipsitch is just on his own crusade. And the incidents that are coming out at various labs are individual incidents, but I think it's not only completely expected, it's completely appropriate for some reporter somewhere, and in this case it was someone at USA Today, to try to do a story getting this whole issue together. And what do? Well, all right, first thing you're going to do is say, how many of these labs are there? And the answer you get when you ask that question of a government agency like the NIH is really disconcerting. I don't know. Honestly, I mean, if you call up NIAID and you say, how many BSL-3 labs are there, they say, we don't keep track of that. Now, put yourself, and obviously as virologists, we understand the reasons for that, but put yourself in the shoes of the general public and say, is that the kind of answer you'd really want to hear? If you look at other stuff that is heavily safety regulated in our society, that's not the way it's handled. If you call up the FAA and ask them how many airports there are in the country, they will quote you a precise number. And they will point you to a website that has terabytes of information about all of those facilities. Similarly, if you call the NTSB and ask how many interstate highways there are, I mean, they will just dump data on you. But the information you get about these biolabs is really spotty and fragmented, and because it's managed by individual institutions, they are inherently reluctant to release it because they don't know exactly how it's going to be used. So none of that is reassuring. And I think it's appropriate that a reporter put in a FOIA requests for all this and did a lot of legwork, frankly. And then, yes, of course, they spun the story to make it a little sensationalized, and the graphics on it, you're looking at a lot of graphics from BSL-4 facilities, even though, of course, the overwhelming majority of these are actually BSL-3s, and it's a totally different situation. But I think I'm not too upset with with Allison Young and Nick Penzenstadler, for the work they did on this. It's the kind of thing that the news media are supposed to be doing.
I think it's fine, but I don't understand the sensational spin. Okay,
right, exactly.
It didn't have to be this way, and I think it discredits it.
Just to give you, I made notes as I went through this thing, and just one example of what I object to is this one paragraph here. Some people find little reassurance that nobody was sickened in the CDC accidents or in the historically low numbers of serious infections among lab workers generally, or that infections spreading into communities and surrounding labs have been rarer still. So what's the fact? The fact is that there's nobody sickened in CDC accidents. There's been very low numbers of serious infections among lab workers, and it's even rarer to have anything spreading into a community. But the way it's spun is some people find little reassurance. Okay,
because they interviewed Lipsitch. Yeah,
this is the Glenn Beck, Rush Limbaugh approach. Now, I'm not saying that Condit is a jerk, but some people think he is. All right. The implication is that Condit is
Especially
a jerk.
Mrs. Condit.
Give me a break.
Yeah.
Just
I mean,
kidding.
yeah, it's because at the same time they're doing this investigation, you have Lipsitch out there making these inflammatory statements, and yeah, they talk to him, and he's one of the few people willing to talk, whereas the folks who would defend these facilities are less willing to go on record, with the exception of Vincent. So yes, I do quibble with some same as Rich. I would quibble with some of the spin that's been put on this. But I don't blame them too much for seeing this as a problem.
Would you go so far as to say that they were fear-mongering at some level?
Not the reporters.
No, I don't mean the reporters. I meant Lipsitch.
Oh, absolutely.
Okay.
That's his shtick. I mean, other than his main job, he that's his hobby, is fear-mongering. And yes, they talked to him, and he spread that to them. Which makes a heck of a story. Obviously, this is a gripping thing that people are going to get drawn into. But I think the underlying problem with it, which they do a very good job of pointing out, is the poor information tracking and the resulting secrecy, which frankly looks really bad.
It's not secrecy. It's just they don't know.
But it's, as I say, the effect is secrecy. Yeah. If you can't track this information and yet you've got these files and then you're reluctant to release them for whatever reason, and I just, I clicked on my state and I looked up UMass, and they tried to charge a hefty fee to release the stuff. USA Today filed a complaint, and finally they got the stuff for free, which is a classic delaying tactic of people who don't want the Freedom of Information Act. Yeah. As I say, if you look at other safety-regulated activities, and it's not necessarily stuff that's risky or dangerous, right? Like aviation is airlines are the safest way you can travel, and if you look at any industry that has safety regulation going on, the records of it are often meticulous and public and transparent, and when they're not, it is totally appropriate for reporters to raise a hullabaloo about that. And I think that we in science need to get our house in order. We need to have a national standardization of BSL-3 and BSL-4 procedures and facilities and transparent record keeping. Well,
here at Columbia, we gave them what they wanted. We just removed locations.
Sure.
Okay, and that was appropriate. We didn't resist. A lot of places did resist. I understand. It's unfortunate. But we're transparent. We're not hiding anything. We have records of everything that goes on here, and we know what's happening. We have a procedure for dealing with everything. And I can tell you, by being on this committee every month, we take care of things.
The thing I'm saying is that those procedures should be standardized, and everybody should follow the pattern of being transparent about them. And I think that would address a lot of these types of concerns.
This would work for the university system, but the private sector might have a problem with a lot of that. And
no proprietary information has to be released here. The private sector companies who are maintaining BSL-3 labs are intimately familiar with government disclosures because they're all FDA regulated.
Well, I wasn't just thinking about the ones in the U.S., though. I was just thinking about, the incidents that have occurred. Marburg virus, for instance, is named that because...
Well, sure. But that's Germany's problem, though.
That was
right?
Germany's problem. That's right. But still, you might get some pushback from those kinds of industries.
But those industries ultimately do disclose stuff, and they are the stuff that they would have to disclose is non-proprietary. Sorry, Kathy.
That's okay. I have two observations. As an investigator whose proposal was before our IBC, I was notified that the minutes at which my proposal was discussed were going to be released because of a FOIA request. So it probably was this or something related. Yep. And kind of a more meta view of this thing is the USA Today thing for me was not easy to do on the web. But now that I'm scrolling through it, I'm just looking, I got to the point where there's a picture of one of the suits blown up and lying on the floor and people looking at it, and it's with the arms outstretched because that's what the suit does.
Yes.
And so it looks like it looks like a dead suit on the floor. That seems to be a little sensational. And also my first thought when I see things like this is almost always, oh, these people are gunning for a Pulitzer Prize. That's just the cynical part in me. They're not really trying to necessarily tell the best story. They're trying to do something that's going to be newsworthy, and in the sense that it's going to catch the attention of.
I think they'd have a better
journalism,
chance at a Pulitzer Prize if they did it in a less sensational fashion.
I think you could equally argue that every paper you pick up, they're gunning for a Nobel Prize, or it's everybody in every industry is trying to make some accomplishment. But
Kathy, we had those suits in our needle video.
yeah,
Oh yeah, no, I know, and I saw them too. But I think when we saw it blown up, it was sitting on a bench.
Yeah,
Yes.
that's right,
It
that's
wasn't
right.
on the floor with people in flip flops and tattoos, looking at it. So yeah.
Anyway, I have no objection to making this transparent, but it's just done in kind of a sleazy way. And, the minutes they have from us mostly concern BSL-1 and BSL-2 experiments.
Sure.
They have it labeled as Ian Lipkin's BSL-3, but it's not just that. It's everything that goes on here. So, if you have there are lots of incidents in 1 and 2 laboratories that go on, but they don't tell you, this is 1, this is 2, this is 3. You have to look at the notes and figure it out yourself. So it's kind of misleading in that way. Now, I know they had a lot of work to do, but they decided to do this, so they should have done it properly. Anyway, I think it's fine that everyone knows what labs are where, and so forth. Just look at it. Take a look at your state and drill down and see what's going on there. Michigan's is pretty okay. They don't say anything. For ours, they say, Lipkin has got samples that may contain Nipah, which killed 100 people. Come on. All right. But while I was looking for Lisa's mail, a follow-up on the MERS in South Korea, I found another follow-up from Todd, which just came in, and I think you ought to read that one, Alan. I pasted it in there.
Okay. Dear Twivers, while listening to episode 339, I was so disappointed that Alan missed a great pun opportunity while discussing the herpes virus paper. Vincent brought up viral host shutoff protein. As soon as I heard it, I was certain Alan would make some sort of VHS joke. Well, I guess we could always rewind to that point.
Ooh.
Thanks for taking a complex paper and putting it into terms that a layperson like myself can sort of follow. All right. Thank you, Todd.
It's a tough paper, yep.
Yes.
We did it. All right. Now we go to our main paper, which is a paper published in Cell Reports. That's the open access journal, right?
Yep.
So everybody could see this. And it's called Mutational Analysis of Measles Virus Suggests Constraints on Antigenic Variation of the Glycoproteins. The authors are Fulton, Sachs, Beatty, Wan, Lee, Palazzi, and Heaton from Mount Sinai here in New York City, Department of Microbiology, Genetics, Medicine. And this is this addresses an issue that comes up very often in my classes, and I think here on TWiV and so
TWiV.
forth.
Yeah.
Why is it that among RNA viruses, which all make their polymerases make mistakes, and they all have error frequencies at about the same level, why is it that some undergo antigenic variation, like influenza virus, rather promiscuously, and others, like measles, does not? There's one serotype of measles, and you got one vaccine that we've been using for 50 years. Why is that? So that's what this paper addresses. So measles.
Good question. I asked this question of myself.
Yeah,
Yes.
of course.
Mhm.
Unfortunately,
how do you get the number of serotypes that we have for these various? Why do we have 3 of polio and not 5? Or
that's the thing. I think we had mentioned this for dengue. Therefore, and then someone wrote in, well,
Right.
there are 5 serotypes. I said, okay, well, they're not dozens and dozens.
Right.
It's not rhinovirus.
In fact, in the end, you will not know the answer to this question.
because it's a why question.
Also, we don't know. We don't know why the result is as it is. We will know a result, but it still doesn't explain it. There's more to go. But
it gives you a way to think about the question, at least.
Yeah, which I sort of anticipated already, but the experiment had to be done.
Yeah.
So this is on measles virus, which, again, one serotype, one vaccine has been helping us for many years. It's a negative strand RNA virus. It's got an envelope around it. The negative strand is about 15, 16 kilobases. 8 proteins are encoded in it. And the way this genome is expressed is polymerase, which comes in with the particle, makes individual mRNAs that encode for the various proteins. And what is this? Okay, you're going to explain this airplane in a bit, right, Rich Condit? I guess after we do the results, right? Because we need to do the results first.
Well, you could actually do it as a preview. I mean, it could do it now.
What is this? You got this airplane picture.
So reading this paper, and this connection will be made as we go along. Reading this paper reminded me of a story that I had heard quite a while ago. That's I think pretty well known, where this guy, let me get his name.
Abraham Wald, a statistician, was asked during World War II to help the British decide how to armor their bombers. Okay? So the deal was that they wanted to put armor in the planes to keep them from getting shot down, but you want to armor them strategically, because if you armor the whole plane, the armor is so heavy you can't carry as many bombs. So you want to do it optimally. And what Wald did was he analyzed all the planes that came back for where they were shot up with bullet holes and told them to armor the planes in the places where the bullet holes weren't.
Right.
Okay?
Right.
Because the planes that survived were the ones that weren't shot in those places. And they did exactly that. And this little graphic that I embedded here is really telling in this, because where the bullet holes aren't is, for example, right under the cockpit or the engines.
Yeah.
Right?
Yep.
There's a couple of strips on the wings here that I don't understand. I'm wondering if there's fuel tanks there.
Fuel tanks,
there are
probably.
Fuel tanks. Sure they are. Sure
How about the tail also? There's a part.
the tail also.
Yeah, the control of the rudder.
The rudder.
Yes, exactly.
Right.
Yeah, okay.
So it's absolutely fascinating. And this paper is the same way. This paper is exactly the same way.
So
let's shoot bullets at the virus and see what places where it doesn't get hit and survives.
Right.
And for those of you for whom that story about the bullets hitting the plane sounds familiar, it might be if you're a Car Talk listener.
Yes.
Because they've had it as a puzzler at least once, maybe twice.
Yes.
All right, so they are able to introduce mutations throughout the measles virus genome. They have cloned DNA copies of the viral genome. You need to use a couple of plasmids that you put in cells to get virus out. But essentially what they do is they mutagenize the genome by sticking in 15 nucleotide inserts using transposon mutagenesis. So with transposons, you put in a plasmid with this 15 nucleotide insert, along with the enzyme called the transposase that's going to help insert that sequence throughout the genome. And they basically get quite random insertions. They have pretty much every possible position gets an insertion, over 10 to the 5th mutants in the end. And that
they do the mutagenesis in vitro on clones.
Right.
So there's no the
bias,
virus,
no selection.
There's no selection or anything. And then if they sequence all those clones and ask for the distribution of mutations, they're all over the place and evenly distributed. So they've made a good library.
So you make this, and you just imagine a DNA copy of the whole genome, and you've inserted 15 nucleotide inserts all the way down the genome, basically at every position.
Right.
They put
And
bullets through every part.
Right.
Right. And
And let's see who comes back now.
the transposon itself is actually longer than that,
Yes,
but then
it is
they
longer.
give
They
it
cut
out,
it out,
and then
yeah.
it leaves a 15 nucleotide
Correct.
piece.
Correct. So then they take this collection of mutants, and you got to put that in the title somehow, Alan, right?
Which part?
This idea of see who comes back, right? Then they put these plasmids into cells and see what viruses come out. They can actually sequence what goes in to determine, the coverage. Make sure you're putting in what you expect. With deep sequencing, you can do this. And then you put in the plasmid collection and you see what viruses come out, right? And what they find is that very specific regions of the genome can tolerate insertions. So they can see insertions in the intergenic regions, the spaces between genes, with the exception of one between the H, which is the glycoprotein gene, and the L, which is the polymerase gene. I'm sorry. Yes, except those. Sites in the N gene, the nucleoprotein, which coats the RNA. The P gene, a phosphoprotein involved in RNA synthesis. The M gene, which encodes a protein that lies below the membrane, they get insertions into those. But the F, the fusion protein, the hemagglutinin, and the L gene, the polymerase, very few insertions in these areas. All right. So I guess those would be the ones that don't come back, right?
Those don't come back. Right. They got shot down.
So we need to put armor on them, of course. That the analogy fails at that point because we're not putting any armor there.
And it's measles, so we actually don't want it to come back.
I do not want it to come back.
So the fusion and the hemagglutinin,
yeah,
those are decorating the outside of the virion. It's a membrane virus. They're decorating the outside of the virion. The hemagglutinin is what binds to the receptor on the cell, and the fusion protein is what fuses the virus membrane with a cellular membrane to get in. But importantly, so those are important functional proteins, but they are also the proteins to which one makes antibody in a vaccine. Okay, that's what determines the serotype.
Right. And they actually do some validation of these data. They reconstruct the insertions into a virus with GFP in it so that they can detect it more easily or with more sensitivity. And they show that, a lot of these areas that didn't tolerate mutation, actually, they cannot rescue any viruses. I think they do get a few, but they multiply at very low titers. They seem to be, have some compromise in their replicative ability. But basically, they show that you cannot change these genes. They have a lovely figure, I think, figure 2, part A, which graphically presents these data really nicely. So they have different colored lines representing every possible insertion along a picture of the genome. And so they show what they put in, which gives you lots of colored lines. And then past this yield of the transfection twice, and you can see at the first passage, you now lose a lot of those lines. You lose a lot of the insertions. And some of them are more prevalent than others. You can see the wider bands of different colors. And then on passage 2, it pretty much stays the same. It's kind of neat that it shows the intense bottleneck, you could say, after the input. Mm-hmm. And I think I have seen these with...
we've seen this somewhere else.
I think in one experiment we talked about, also from Sinai, where they put various siRNAs into a viral genome.
That's right.
I think they expressed it in the same manner as well.
So now that you've described this, I finally understand this graph. I puzzled over it for a long time, but I get it. Because I can see these initial mutations going away.
Right. Exactly. So that's really the story, is that these glycoproteins on the surface are rather rigid, architecturally rigid. They will not tolerate, these are 5 amino acid insertions, right? Now,
this is a pretty big change.
I wonder if you would see anything different if you did. Well, clearly, those 2 genes are different from the rest of the viral genome, right?
Right. Other genes can tolerate these insertions.
Maybe
except
if you did,
L.
Except L. Maybe if you did a 1 amino acid insertion, you might see the same overall pattern, but maybe you could get more viable viruses with insertions there. All right. So that's the observation, that this virus is constrained in the H, the N, and the H, the F, and the L proteins. And the L is the polymerase, which I suppose you can't mess with,
right,
in any way. It's just evolved to be perfect the way it is, and no, we're not tolerating any insertions. But why are the glycoproteins so constrained? And influenza virus, by the way, I didn't mention this. this group did a previous similar study with influenza virus, and the HA will tolerate insertions of the type that were done here, but only in the globular head of the HA molecule. So again, the HA is a glycoprotein on the virus surface, right? It's got a fibrous stalk to which is attached a globular head, and that head is known to undergo extensive antigenic variation, and indeed that head could tolerate these 5 amino acid insertions, but the stem could not.
And the stem is less prone to those kinds of. I mean, it's more
stable.
Universal. It's more stable.
Yeah.
It's what people are trying to direct universal vaccines to.
Exactly.
Right.
And that malleability in the influenza head is what gives influenza the antigenic drift. Okay, and so in some ways gives it, if you like, on an evolutionary scale, a selective advantage.
So they compare this mutability, if you will, with influenza, measles, and 2 other viruses, hepatitis C virus and Venezuelan equine encephalitis virus. So in the influenza, the NA and the HA surface glycoproteins can tolerate insertions. Hepatitis C virus, only a very small part of the glycoproteins can tolerate insertions. Venezuelan equine encephalitis, the glycoproteins can tolerate insertions, but not measles virus, as we saw in this paper.
Can I ask for a clarification?
Yeah.
When the transposon inserts the 15-mer, is it the same 15-mer in all the mutational sites?
Yes.
Yeah, it
Okay,
is.
so if you were doing this as a thesis problem and you gave a student one of these proteins and you said mutagenize it and see if it is affected, wouldn't you break it into its component segments and try different amino acid substitutions rather than 15 at a time? Isn't that what you were saying before? Maybe single amino acid
shorter ones,
substitutes would work, whereas 15 in a row may be too much to ask of the system to give you.
Well, it's 15 bases. It's 5 amino acids.
Okay, but it's still 5 is more than 1.
Yes.
I have to disagree with you guys. At least my interpretation of it from Table 1 is that the inserts ended up being all different.
All different, yeah.
And I went through the molecular biology as best I could, and I couldn't quite figure it out, because in the supplemental methods they talk about the 15 nucleotide transposon, but then they give the transposon the insertion sequence, and it's only 10 nucleotides. It's the not one sequence with a nucleotide on either end. So something was missing that I just couldn't deal with.
They are different, yeah. But still, the size issue is a good question. Although I suspect if you did one amino acid insertion, the overall result would still be the same, right?
But maybe the antigenicity would change, though, if you did that. I mean, isn't that what they're looking at? The vaccine depends upon a stability of antigen. So if you did a single amino acid substitutions all the way down the length of each of the 6 proteins, maybe nothing would happen except the antigenicity might be different.
Well, I think that maybe, but I think the implication of this is that would not happen, because the implication of this is that this thing does not undergo antigenic drift, because even small changes this is an extrapolation, even small changes in these proteins would not be tolerated.
Right. And if it were capable of drifting antigenically, that should have happened.
Right.
So just another naive question, of course. In a measles outbreak, without the vaccination advantage in a population, is 100% of the exposees infected? Or are there groups that are not because they don't have the proper ligand for the receptors? Well,
in any outbreak, you never get 100%, right?
But measles is pretty
but I still think there are people who don't get infected, but we don't really know why.
I mean, I was just wondering whether nature has already answered this question someplace, and you could find a group of people not susceptible because.
Yeah. Well, nature has given us the data. They say we've been using one vaccine for 50 years, and we don't get any escape, whereas we have to change it very frequently for flu. But why this protein is so constrained, we still don't know.
One of the things that they say in that regard is that flu, for example, its receptor is just a sialic acid, which is pretty simple. And the receptor in this case is I forget what it is, but it's much more complicated.
It's a protein, right?
Right,
So,
and same,
it's a protein.
same for other viruses where we know the receptor and there's a limited number of serotypes. It's frequently a specific protein receptor.
So that interaction is going to be more complex than just glomming onto a sialic acid.
So why does changing the knob on the influenza not affect the way it binds to those cell then?
Yeah, that's a good question. I guess, sialic acid's a small molecule, so these changes don't have much effect. But, you can make changes in the sialic acid binding pocket, and that affects attachment for sure. So, but the antigenic sites are a bit away from that, so I don't know if that's the right answer. You could argue that it's not correct. But certainly experimentally you could look at that in some ways. Anyway, so I come out of this. It's a cool paper. It's a neat approach to this. But in the end, I know it's a why question. But why is this architecturally constrained, but the influenza virus is not? I mean, we draw the measles hemagglutinin protein with a stalk and a globular head, right? Why is the globular head constrained?
And how is measles not figured out the same trick as flu of binding a less specific receptor?
Yeah.
To give it that flexibility. Apparently it's just not been evolutionarily selected for.
It hasn't been.
Or maybe it's in an evolutionary space where it can't get there from here.
Right. That's right.
I think it suggests that there's something about the H and the F that maybe the way they have to interact. I guess you've already said that. I was thinking, interacting with each other kind of in sequence or sequentially in the infection process, or something that it's just not going to tolerate the changes.
Could you make a measles virus with a flu HA?
Would that be a gain of function
Yeah, it would
experiment?
be.
Yeah.
Sure.
No, you can't do that.
No, I can't. But let's say it was infectious, such a virus. Would that hemagglutinin then be unconstrained, and would that virus then undergo antigenic variation?
I'll bet you it would.
Unless it's, as Kathy suggested, maybe it's an interaction on the particle with the F protein that is constraining it in some way. But flu has HA and NA, so that's kind of I don't know. Anyway,
and of course, to do that, you'd need to muck around with the rest of the packaging for measles so that it could actually complete more than one cycle at that.
I'm not sure anyone would let you do that experiment.
No.
That's unfortunate because it's a good experiment to do.
You could do it. You could do it in a packaging line so that the virus can't actually replicate outside the line.
Yeah, maybe.
Mm-hmm.
I mean, it may in fact have been done. Any measles people out there? Maybe Paul Dupre. Has anyone ever tried to substitute the glycoproteins? Let's see what you think. All right.
I wanted to point out that in doing the analysis of the other viruses, they took advantage of data sets that were already out there and analyzed those. So they didn't do this. They didn't have to do the mutagenesis again.
Right.
They also have a graphical abstract that I thought could have been done a little bit better. For instance, they have 3 potential genomes with mutations indicated in them, and the one at the bottom corresponds with the virus at the top. And then it's just not as good graphically as it could have been for a very simple, helpful graphic.
Who makes these? Do you know, the authors or the journal?
I don't know. I thought the authors did.
Probably, because they vary in style, from one paper to another.
Yeah, but clearly the color in the virus is supposed to represent whether it was mutant or wild type relative to the red insertion of the transposon. But it took me a while to figure out. And it was after I read the paper that I looked at the graphical abstract, and I thought, I understood the paper better than this abstract.
So, Rich, if you did this with vaccinia, if you did transposons all the way down the genome, would there be any constrained regions?
Oh, yeah. So that's actually a really interesting question. And I'll bet you from what I know from what Kathy has said, the answer is going to be the same for adenovirus and a bunch of others. In the case of vaccinia, if you take all of the orthopox viruses and even all of the pox viruses and line them up and do sequence homology, you find a conserved region in the middle of the genome where all of the sort of fundamental I hate to call them housekeeping because it's such a, but, housekeeping genes are, polymerases and structural proteins and that kind of stuff that you really can't tinker with. And out on the ends of the genome are all of the immunomodulatory stuff. And that, even in nature, that's highly variable and gets tinkered with a lot. So what you would probably find is that the middle of the genome is pretty intolerant of insertions, and out towards the end you can tolerate quite a few more, especially if you're doing it in culture. You'd get a different answer if you were doing it in culture than if you were doing it in an animal. Not an entirely different answer, but a slightly different answer. As a matter of fact.
Yeah, right, because those genes you need for immune antagonism, for example, you wouldn't. That would be variable in culture.
That would be a very interesting experiment to do in an animal to try and get an idea of what sort of hierarchy of immunomodulatory genes were in terms of importance.
Right.
That's a nice idea. It'd be a tough experiment, but nice idea.
I'd like to do it with polio as well. I bet the capsid would be constrained because there are 3 polio serotypes,
Right.
but some other proteins might not be. I always get great ideas from papers, but you can't do them all.
Well, and
Right.
an unenveloped virus would also have the problem of maintaining the structure of the capsid.
That's right. That's right. All right, let's do some email. Kathy, can you take that first one, please?
Sue Ellen writes, I listen to your podcasts, all 3 of them, every week, and I just love all the science. Vincent, can you please move your cursor?
Yeah, sure. What does it do, actually, when it's there?
Puts a little flag over the next words, and I couldn't quite interpolate what they were.
Right.
I am pro-vaccine, but of course, like just about everybody else these days, I have anti-vaccine friend. One posted this on Facebook. Amount of mercury allowed in drinking water, 2 parts per billion. Amount of mercury in a vaccine, 51,000 parts per billion. Now, I just know this can't be true, but I don't know how to respond to it in a responsible, science-based manner. What are the facts? Can you help me correct this? And I sure hope it's wrong. I looked online, but I can't find official numbers from the industry, only crazy numbers from the anti-vaccine people.
Isn't that sad that you can't find anybody?
Right.
Oh.
But
Well, it's because it's so far away from fact that the industry hasn't. I mean, there's no. you can't address this on a fact for fact basis.
Yeah, right.
so my first thought was the fact that we've heard from Harpocrates before. It's a person who has posted on the amount of formaldehyde in vaccines and aluminum, I think.
Yeah.
So I wrote to him right away and said, did you do one on mercury? And he wrote back and said, I think that part of what they're talking about is methylmercury, which has a limit of a certain number of 0.001 milligrams per kilogram per day. But ethylmercury, which is in the vaccine stabilizer thimerosal, has a half-life of around a week. And then they're confusing something that people are exposed to multiple times every day, drinking water, to a one-time acute exposure, a vaccine. And then, as he points out, and we're going to hear in more detail in a minute, only some flu vaccines contain thimerosal at the present, and I think that's the only vaccine that has thimerosal.
That's right.
But then Vincent wrote to Paul Offit, who sent us his book with a whole part of a chapter about this. So
What's the title of this book?
Vaccines and Your Child, Separating Fact from Fiction.
Yes. So he wrote this with Charlotte Moser, and after he sent it to me, I recognized it because I did a review of it for the Columbia University Press.
I've seen it on your shelves.
And it's on my shelf there. They had sent it to me to look at, and I liked it. I thought it was a good book for the general public. Yeah, so he has a few pages on, it's a section called Do Vaccines Contain Harmful Preservatives like mercury? The preservative in vaccines that has caused the most concern among parents is thimerosal. And he goes on to explain why thimerosal was originally included in vaccines. And then
I think that's worth pointing out. It's because when there were multiple dose vaccines in the early 1900s, people would go in and out of the same vial multiple times to give that vaccine. And if you were one of the people getting the last vaccine in the vial, there was a higher risk of you getting something else that had been introduced into it, some kind of bacterial contamination or something. So
Right.
people started adding these preservatives.
Right, which remained standard in many vaccines for all the way up into the 1990s, I think. In multi-dose vials. This is a preservative that's only used for multi-dose vials, which are a much more economical way to distribute vaccines.
So he points out that as health officials added more vaccines to the routine schedule, children were getting more and more mercury. So the American Academy of Pediatrics, the U.S. Public Health Service, in 2001 decided to remove thimerosal from all vaccines, with the exception of multi-dose preparations of inactivated influenza vaccine.
Which, by the way, was not based on any actual identified risk.
Right, that's correct.
That was based on a little bit of public panic and a misinterpretation of some old data on methyl mercury, which is a completely different compound. And if you're confused about the methyl versus ethyl thing, it's like the difference between methanol and ethanol. Ethanol gives you a little bit of a buzz when you have a beer. Methanol causes you to go blind. So the methyl mercury that everybody's concerned about is a radically different compound from the ethyl mercury that is in thimerosal.
So he writes, unfortunately, the demand for rapid removal of thimerosal caused some parents to wonder whether it had caused harm, specifically autism or subtle forms of mercury toxicity. Because mercury at high doses can be toxic to the nervous system, this concern was reasonable. And then he goes on to talk about facts about mercury. It's part of the Earth's surface, released by burning coal, rock erosion, volcanoes, settles into lakes, rivers, and oceans, converted by
tuna sushi,
converted by bacteria to methyl mercury. We had sushi now in 4 straight episodes.
That's right.
Methyl mercury is everywhere. Fish we eat, water we drink, infant formula, breast milk. There's no avoiding it because everyone drinks water.
Fillings.
Everyone has fillings.
Yeah, and your teeth.
Oh,
They
yes.
used to eat it. There's there also.
Everyone has
fillings.
small amounts of methyl mercury in their blood, urine, and hair. In fact, a typical breastfed child will ingest 400 micrograms of methyl mercury during the first 6 months of life. That's more than twice the amount of mercury than was ever contained in all childhood vaccines combined.
I think that fact is worth repeating.
Yes.
A typical
Really good.
breastfed
No question
child
about it.
will ingest more mercury than was ever contained in all childhood vaccines combined.
Right.
And that's methyl mercury,
Yes,
right? Which is worse than ethyl mercury.
far
Right.
worse.
Yes.
Right.
Yes.
And because the type of mercury in breast milk, methyl mercury, is excreted from the body much more slowly than that contained in vaccines, ethyl mercury, breast milk mercury, is much more likely to accumulate. That doesn't mean that breast milk is dangerous or that infant formula is dangerous. It means only that anyone who lives on the planet consumes small amounts of mercury all the time.
So tell your friend on Facebook that breast milk contains more mercury than the vaccines their kid is getting.
Yeah.
That'll pose a thinker for him.
That's right.
Yeah.
And by the way, there wasn't one single Mad Hatter out there that suffered from autism.
That's true. Yes, that's another issue, is that the whole autism thing is a red herring, because that is not a symptom of mercury poisoning. It never has been.
That's exactly.
Produces an entirely different pattern of symptoms.
Yeah, quite right.
And but just to be careful about that fact, it's the amount of breast milk in 6 months being twice as much than was in the childhood vaccines combined.
But it would be fair to say that if your infant is breastfed, they will consume more mercury in breast milk than if they got only mercury-containing childhood vaccines.
Exactly.
Which is not going to happen anyway.
Which is not going to happen, and they aren't even available
Which
anymore.
also tells you something about the amount of mercury in our diet, because that's where that's coming from to get into the breast milk. So,
Right.
Come on.
All right. Alan, do you want to take the next one, please?
Sure. Peter writes, Hi, TWivers. Not sure if this recent Nature Communications paper is within the remit of TWiV, as it is immunology rather than virology, but I think it could be worth a mention. We're all about very precisely defined turf around here at TWiV. The authors highlight many seasonal changes in the immune cells, immune and inflammatory responses, with a more inflammatory response predominating in winter months. It was also found that the antibody responses to several vaccines were influenced by the month in which vaccinations were given. The paper is open access, but I found it rather heavy going and feel I would gain more from a discussion of the paper by people with a better understanding of the subject than me. And links to this Nature Communications article. Everybody has an open access journal now, and this is Nature's open access journal. Widespread seasonal gene expression reveals annual differences in human immunity and physiology. And first author is Joaquin Castro-Dopico, and senior author is John Todd. And I have not had a chance to read this.
So they basically look at RNAs, probably by RNA-seq or something like that, in different, so white blood cells and adipose tissue, and different people in different seasons throughout Europe and Oceania. And they find that the levels vary.
Of course.
And, how much and how significant the variation is anyone's guess, right? I mean, you can see numbers here. They don't look they don't look huge to me, but there's clearly a seasonal variation if you look at specific genes, right? So let's see. They have GAP-DH as their control in one of these figures, and that's constant. It doesn't seem to vary at all with the month of the year. And in contrast, a gene called ARNTL, which is obviously an immune response gene of some kind, has a peak in July. So lots of these genes, interleukins, cytokines, and so forth that they look at have seasonal variability.
Do they control for where you live, of course? They say winter, but if you live at the equator...
They do, yeah.
There's no variation in people who live at the equator?
I don't think they looked at it.
Well, they looked at some Ghanaian population for some part of their figures, but mostly they're taking into account when winter is in your respective hemisphere.
I just wondered if it was an internal clock making this happen or exposure to organisms which have seasonalities to them.
Well, that's a good question, right?
That's a good question.
We don't know which is which. Cause and effect.
There you go.
Yeah, I like that question. That's good.
Or someone with this pattern moves to another region which doesn't have as dramatic a season, or it moves to the southern hemisphere. Does it reverse, or does it stay the same?
I would assume so.
It depends on why it is there, though, right?
Yeah, I think it's
diurnal
difficult.
rhythms tend to remain the same regardless of where you move these things. So, for instance, aplysia, which is a sea slug studied here at Columbia University, comes from California, and the tidal memory of an aplysia is built into its responses in its diurnal rhythms, and they maintain those same rhythms here as they do on the West Coast.
So they're
imprinted, and then they stay with them. So I just wondered whether this could be something similar.
So the Gambian cohort also had variation, right, Kathy?
It did.
You're right. It's Gambian. I don't know why I said Ghanaian. Yeah.
So they, what was I going to say? So a lot of the genes we've talked about, TLR7, IRF7, those sorts of things, RIG-I, MDA5, these vary, to give you an idea of the genes they're looking at. They say here that, the mechanisms remain to be established. Daylight, ambient temperature are cues that could coordinate seasonal hormonal phenotypes and cell fate decisions.
Male, female.
Diurnal entrainment of the circadian clock.
Yeah.
But they say at the end, these, regardless of the mechanisms, they could explain seasonality of human disease. Although you would argue, Dickson, that is the inverse, right?
Yeah, well, I wouldn't argue. You need more data to say whether it is or isn't.
I'm just not sure what they're... I know what they're measuring, but I don't know what they're really measuring.
Exactly.
The immune system's connected to everything in the body.
You bet.
And I would be shocked if you didn't see seasonal cycles like this. It would mean that you hadn't done the assay that you wanted to do. Of course, you see seasonal cycles because metabolism cycles with the seasons and everything to do with it. And of course, the immune system is affected. And of course, there are going to be differences in all these markers related to the immune system because it's affected by this same, if you will, tidal force of the seasons. But what that means, I don't know.
Yes.
Be interesting
and
to
whether
see. No, go ahead.
And as you said, Dickson, it's unclear whether this would predispose you to infection or infections that spread preferentially in the winter predispose you to these cycles or some underlying factor predisposes both. I don't know.
I have a great experiment here, by the way. That is to send about 4 of those people up to the International Space Station for a year and monitor them every week and see what happens. I'll be one of those people. Rich, I know Rich will be the other one.
Yeah, I'm ready. That's good. I'll give blood for that one.
There you go.
That's a good idea.
Yeah, I like that idea.
Yeah.
Rich, can you take the next one? Reed?
Sure. Reed writes, Dear Vincent and friends, I came across this recent article in this week's Nature and was curious about TWiV's team's opinion, especially regarding ideas about new policies for antibody production and validation. And he gives another related article. Says, how feasible do you think these suggestions would be? How much of a reproducibility problem would be fixed? Thanks for your time, Reed. And the article that he lists is an article in Nature about reproducibility of antibodies. So this, basically the bottom line is that the estimates are that as much as half of the commercial antibodies on the market are junk. Okay.
Or not as specific as there's.
They're not as specific as they're supposed to be, not as reproducible as they're supposed to be. They might be getting a wrong protein. There may be a lot of background, et cetera. And it goes off on people wasting years and hundreds of thousands of dollars doing experiments with antibodies that turns out we're looking at the wrong thing or something like that, and says that this is a problem. And there are those who would advocate some sort of standardization of the production of antibodies. And the second link that it's mentioned in the first link, but the second link that he put in is it's a letter, I think. Let me get it up here. Also in Nature, by Andrew Bradbury and Andreas Plukson, suggesting that there be a standardization process where commercially available antibodies should all be sequenced recombinant antibodies. Okay, so any particular antibody that you're going to sell commercially, you have to have the sequence of the antibody encoding gene, and it has to be in an appropriate, probably a standardized cell that's producing that antibody, so that would so you would know exactly what you got, and it would be entirely reproducible. Personally, I don't think that's a practical solution for this.
Yeah.
Okay,
I thought a lot of this was monoclonal antibody anyway.
Yes, there's not always, but a lot of them are monoclonals.
So how can that be variation then? Because isn't that all clonal?
That's a good question, but I can tell you that you can buy an antibody, and on the
it's
website,
a dud. Yeah, I know.
They show you a lovely Western, and you get it, and it doesn't do anything.
Yeah, you're right.
Right. And of course, there are many explanations for that.
It could
but
degrade,
we have wasted many thousands of dollars.
Oh, me too. I wasn't that.
I would love if they could be all cloned and really very specific.
Yeah.
But I agree. These companies are in this to make money, and they probably make a lot of money doing. The way they do it for many of them is to make a peptide and stick it in rabbits, right?
Right.
And they charge you for maybe 100 microliters, $300-400.
And do they submit their own analysis of the specificity of the antibody, like a Western?
Yeah, they have. You go to their website, you can see a Western blot, and then you do it, and you don't get anything.
Yeah, right.
So really, what this
you get an Eastern.
The practical solution to this, in a way, and there's some suggestion in these articles that to some extent this is already happening, is to let the free market take care of this.
Yes.
Right. So that in order to make money selling antibodies, you have to sell a reliable product, and the people selling the best product will be the winners down the road, and that will take care of itself. It hasn't so far.
Yeah,
but
I don't think it's going to. They sell so many different antibodies. They can sell enough reasonable ones, GAP-DH, to stay in business.
Some kind of actin.
Yeah.
Right, but, first of all, I love this quote in the article. One researcher who says, I wasn't trained that you had to validate antibodies. I was just trained that you ordered them.
Use them. That's right.
Very good.
Which, it says this on the vial, and of course in the lab you're used to ordering restriction enzymes from New England Biolabs, and they show up and they cut where they're supposed to, and then you order an antibody from somebody and it shows up, and you do your experiment, and
you trust the result. Yeah,
when it doesn't
trusting
work,
in the reagent.
It doesn't work. You order one from another company till you find one that works.
If there is one from another company,
Yeah.
but
And then
in
you-
the
and then
meanwhile,
you
you
hope.
talk to the technical people at the company, and they say, "Well, you got to try this," and
Right.
and you ask, "Could you send us, a sample of your extract that works or your positive control?" No, we can't do that, but you have to do this,
Yeah.
and this, and
Kathy, you
before
just
they'll
said it,
even
but
give you a refund or send you a new aliquot of it.
Yeah, your positive control, that's absolutely critical. If they don't send you the antigen along with the antibody so that you can do the Western and get the exact same result that they got, then that should be one of the ways of testing these products out.
But
Yeah.
they don't.
But they should.
Usually
It's
it's
not
a
the
cell
industry standard.
Usually
Yeah.
it's a cell extract anyway that they're using, so there's no antigen for them to send you. And I wouldn't want an antigen. I want a cell extract because that's what you're going to do
Right.
your experiment.
Yeah.
Okay.
Yeah, and the thing is, if you find a company that sells an antibody that gives you the result you want, you are simply hoping that they are not going to change anything about their manufacturing process
Sure.
or get a
That's right.
contaminant or have any other kind of a problem that ends up
Batch-to-batch variations.
shipping you something else.
That's right.
Maybe it was a fluke that it worked.
Well,
Yeah, if
we,
it's a polyclonal, next time they make
Sure.
it, they may not be able
That's
to
right.
reproduce it.
we
That's
have had
black
it.
magic.
We have had from the same company a batch that works great. Next batch
Yeah,
doesn't work.
or it degrades.
Yeah.
All that
Yeah.
stuff is possible. Yeah.
Really,
Yeah, I do think this is something that the market could fix. If companies did what Amazon does and put product review options on their websites.
Great idea, Alan. That's
Right,
a
so
great idea.
I bought this antibody and it's useless.
Sucks. That's right.
Star.
Terrific. I like that.
Yeah,
that.
Yeah, that's a good idea.
I
You
do
could read
like that.
the reviews, or I used to use this and it stopped working. I don't know what they did to their batch process.
Yeah.
Now, of course, companies would have a motivation not to enable such a feature on their sites, but if one company does it, then the others who don't will be at a significant disadvantage.
And they have an example in this article of one company that went through and reevaluated all their antibodies and threw 30% of them out and kept the good ones, and in fact their business went up.
This is a case where, yes, the companies who make these could very easily beat the tar out of their competition by one company coming forward and taking advantage of this and saying, we're going to do it right. And then that'll be the company everybody buys their antibodies from.
Yeah, I really like the product review idea. That's a really great idea.
Yeah,
I think
good.
that would go a long way. Yeah.
Good stuff.
So there are lots of antibody review websites. I just looked for some, third-party antibody comparison sites. Just type that in
Okay.
and you'll find it. I don't know how many reviews there are, but they're certainly out there. And I'm sure you can find some information, right?
Well, are the comparison sites just telling you who sells something to this antigen and it's raised in rabbit or mouse or rat or whatever? Or are they actually...
There's supposed to be comments from users
Ah,
saying whether
okay.
it works
Oh,
or
that
not.
could be
you know.
useful.
It'd be best to have it on the manufacturer's website, but independent sites
yeah.
could do it too. Yeah.
I could see where the manufacturers might not want to do that.
But again, if one in the industry wants to be a leader in this, they could absolutely clean up.
So another approach would be to submit, if you're doing it on tissues, to have the manufacturer send the exact section of tissue that they use to test it out. It has to work on that section, otherwise you don't get it to work. Because what's your positive control? You don't have any.
Right.
The thing is,
How could
though,
you do an experiment without a positive control?
If you have an antibody that works, it's not so hard to sequence it and clone it, and then you always have great antibody. I don't know why the companies don't want to do that. Obviously, they're making enough money that they don't need to do that.
Right. The market has to send a signal to the companies that you're not going to put up with this anymore.
And that was not a pun intended, I presume, by you, Alan.
Right. No. Send a signal.
Sending a signal
That was just incidental.
companies. Yeah, thank you.
I'm going to take this next one from Katie, and then Dickson, you can take the final one from Judy if you want to
Okay.
look at that and be prepared.
I'm going to practice my reading skills
Excellent.
right now.
But do it in your brain.
I'll do it.
Katie writes, Dear
brain?
TWiV team, I'm a PhD candidate at Cornell University studying co-infections between plant viruses, and I wanted to provide some information about Prunus necrotic ring spot virus and cherry leaf roll virus. Oh, it makes me hungry. If no one has yet written in, I'm a bit behind in my podcast listening since I only listen when doing lab work alone. The epidemiology of both viruses relies heavily on pollen and seed transmission, to the point where Prunus necrotic virus has no known insect vector. Prunus necrotic leaf spot virus is an ilar virus that packages its segmented tripartite genome into single capsids. Like many Nepoviruses, cherry leaf roll virus packages its bipartite RNA genome into separate capsids. RNA1 and RNA2 are packaged into B and M particles, respectively, and both are needed to infect the plant. While there is some evidence that certain strains of cherry leaf roll virus can be transmitted by nematodes, a 2006 paper in the Journal of Virology by Rebenstorf et al. Provides evidence that this is unlikely to be the major form of transmission. The authors examined a number of strains isolated from different host species in several countries around the world and found that the population structure of cherry leaf roll virus is strongly correlated with host species rather than country of isolate origin. The majority of neoviruses like cherry leaf, maybe that's Nepoviruses, cherry leaf roll virus are seed and or pollen transmitted. So isolates are very likely to be transmitted within a host species. One way that cherry leaf roll virus and other neoviruses may be able to survive with separately packaged genomes is that ovules can likely hold more virions than could be transmitted by an insect vector, such that the probability of only containing one virus particle type is low. Remember also that plant virus particles do not use cellular receptors to enter cells, and RNA is actively transported between plant cells through plasmodesmata, cytoplasmic bridges between plant cells. So this is cool. We were talking last time about how you would get both particles into a single cell, right, to ensure that both genomes are there.
Mm-hmm.
And here, we were talking about nematodes and so forth. She said probably not. It's in the ovules.
It's an STD.
It's an STD.
It's germline infection. Yeah, that's right.
And it's Nepoviruses.
Nepo.
Nepo.
She left the P out twice. When she did it twice, I thought I might.
That seemed like the consistent one, yeah. Right.
I find all of this infinitely fascinating and would love to hear more TWiV podcasts about plant virology.
Yeah, me too.
Living in Ithaca, New York, I'm often extremely jealous whenever Rich gives his weather reports, but today is a beautiful 63F 17C day with clear skies. Thanks for all the work you do to put out these podcasts. Mm-hmm. Cool. That's a very cool story, Katie.
Yeah, very nice.
Yeah.
Neat.
And we will make great efforts to have more frequent plant virus. I have one on the queue.
It's very, very important in terms of world economics.
Yes.
All right, Dickson, take the last one, please.
Judy with an I writes, and then she lists an NPR selection of a selection. So NPR is selecting this, and they selected a map of the United States by the cause of death from each entity per state. And so, in other words, you get a map of the United States, and it says, in Texas, for instance, she says, the most common cause of death in Texas is tuberculosis. Really?
That's so weird.
That is weird, but they have a lot of immigrant workers.
It says distinctive, though.
It's not, Judy misread it.
Yeah, it's distinctive.
She's
right,
saying
okay.
the most common cause in Texas is TB, and but it's...
In other words,
what distinctive means in this context.
There's a background of cause of deaths common to all states, and then there are nuance, and in this case, tuberculosis for Texas.
Right.
And so then she also asked, and why are people in Utah dying from undetermined events and their sequelae? Huh? That's a good question, but of course no answer. And then here in California, we die from hyperplasia of the prostate. So I guess women will live forever, with an exclamation point. Thanks for all you do. And Judy is a retired science teacher in San Diego, and she also goes on to say the weather here is cloudy, cool, high in the 60s, calm winds, 62% relative humidity, a wonderful, relaxing day.
These are
Right.
distinctive, right?
Right. Yeah,
right.
And here my geography is really rotten. It's failing me. There's two here with V's where the distinctive is discharge of firearms, undetermined intent.
Yes.
What's that? Is that Arizona and what's above Louisiana?
Arkansas.
Arizona and Arkansas.
So
Arkansas.
those are hunting accidents?
I guess.
I don't
Right.
know.
So distinctive. I saw this before the show, and I looked at the map, and I thought that can't be the most common cause of death in Texas. Is tuberculosis.
No.
So this
lack of proper health care facilities.
Demographer, I think, took nationwide causes of death, and there are something like the CDC tracks like 113, or he calculated mortality rates for 113 causes of death. And then nationwide, there are averages of these.
Sure.
So this is what's the most common nationwide. And then he picked out the cause that varies the most for each state from the nationwide average. So what's most distinctive within that state.
Sure.
So
if you want to die from gunshot wounds, you could probably move to Arizona.
Or yes.
Arkansas. Or Georgia, if you're a black male between the ages of 16 and 25, because there's a lot of murder that goes on.
Here in New York State, you would die. The unusual or distinctive cause of death is inflammatory diseases of female pelvic organs. Huh. So here in New York, men will live forever, I guess.
That's right. Mm-hmm. That's right.
And in Massachusetts, it's other and unspecified events of undetected.
Joining the Utah contingent.
What do we got in Michigan, Kathy?
Atherosclerotic cardiovascular disease, so described.
Huh.
That's because everybody sits indoors and eats chicken wings all winter.
Yeah, they should have done it separately for the UP and the lower P.
This is true. This is true.
You got HIV there in Florida, Rich.
I knew that.
Really?
No, actually, doesn't surprise me. Put it that way.
Okay. All right. Let's.
And in Washington D.C., it's foot-in-mouth disease.
Foot-in-mouth. Dickson, you're making a joke.
I tried.
Let's do some picks. I bet you don't have one, Dickson.
I bet I do.
What do you have?
But I might have picked this one before. I just got back from Singapore. So there are two marvelous facilities in Singapore. One is called a cloud forest, and the other was called Flower Dome. And they're both enclosed, and they're both air-conditioned, which is an essential, because if you want to see a good tropical rainforest, it's right in the middle of Singapore, but it's 95 degrees and 80% humidity, and you can't last for more than 2 hours in those conditions unless you live there. But I went to those two facilities. That was my second visit to both of them, and I highly recommend them. They're absolutely stunning, beautifully done, incredibly scientific. All the plants have names. There are baobab trees in the cool, dry climate dome. Lots of interesting things to see. Not crowded. Take your camera. They allow tripods. It's a fabulous experience. And I was there again, and I had an equally great time.
Do you have a website? We could.
They probably do.
Flower Dome does.
They probably do. Yeah. Both of them. They're connected to each other. They're connected to each other. They're fabulous. It's a big trip, I must tell you. It's a long way to go, but it's worth the price of admission.
Did you go to the toilet where the water goes around the other direction?
No, just sat there because I was right on the equator almost. There was no Coriolis effect.
Thank you, Dickson.
You're welcome.
Alan Dove.
Okay, I have. This has kind of been a running theme. This is the. I guess this is the official "What the heck do we do about this mess" website. It's called Rescuing Biomedical Research, and it is. This is being put together by the same cast of characters who've been commenting on this whole sustainability and the post-apocalypse and the whole mess.
Post-apocalypse.
So the authors on that, Rescuing U.S. Biomedical Research from Its Systemic Flaws paper, Bruce Alberts, Mark Kirshner, Shirley Tillman, Harold Varmus. They are on this, and they have put together this website to try to describe the problem and collect input and get people together to work on solutions.
Cool.
Wow. They have all these areas where they want your input.
Yes.
Compliance, hyper competition, errors. Can we just put TWiV episodes in here?
Why not?
That's one of the things that needs to be fixed.
Arg.
What, TWiV episodes?
Right. Oh, you mean just cite sources?
Yeah, because we've talked about a lot of this.
Cross-reference them.
Yeah.
Oh, that's cool. Well, that's nice that they're doing this.
The workforce to the funding, all that.
Okay. Rich Condit, what do you have?
I want to plug the Virology 60th anniversary issue. So Virology is probably the original virology specialty journal. and it's celebrating its 60th anniversary. And in order to do so, it asked all of the senior editors to invite reviews from various individuals, and the result is a fairly eclectic collection of reviews in virology from, I think there are appropriately about 60 of them, covering a wide range of topics. So I solicited 4 of these, and the other editors solicited their bunch. It is, for the occasion, open access, so anybody can access any of these and have a look.
Nice,
it's quite an interesting collection.
Where was that first published from? Where was the editorial offices?
Where were the editorial offices
for Volume 1?
Duke?
Wow.
Or Rockefeller?
Was it Rockefeller? You
Was
sure?
it Rockefeller?
No, I don't know. It might have been.
I don't know.
Because
and
there
I
weren't
forget
that
who
many
the original
virologists in that.
editor-in-chief was. I should look up while you guys
I
are
thought it
doing
was
this.
Bill
I'll look up
Yachlik,
the
but
history.
maybe that was JV.
No, he was not the original editor. Let me plug one of these reviews that you guys would like. One of the reviews I solicited is from Eugene Koonin.
I was just looking at that, yeah.
Virus evolution,
that's
and
good
it's
stuff.
fabulous. It's really a nice
All right, here's the
article.
history. Started in 1955 by George Hurst, who was
There you
here
go.
in New York, Lindsay Black, and Salvador Luria.
There you go.
Salvador.
Cool.
The earliest English-only journal to specialize in the field. I don't know exactly where
Yeah.
it was founded, but those were the founders.
Interesting.
Interesting.
Yeah.
Yeah, there's a good history here on Wikipedia. Hearst was editor-in-chief for 21 years. Oh, my gosh.
Wow.
And after him, he had 3 successors, Wolfgang Jochlik, Robert Lamb, and Michael Emmerman is the current one. It's kind of like
Wow.
CEOs of Microsoft. There have only been 3.
Yeah.
Right.
Wow.
It's pretty rarefied.
Bill Yachlik was editor for a long time. Long time.
Got to put that link in the show notes. That's a cool
Yeah,
one.
Thank you.
Okay, Kathy, what do you have?
I picked a blog post from the White House Office of Science and Technology Policy on the value of basic research, posted by Jo Handelsman. As we know, she's a microbiologist. And it's interesting because it talks about some things that were previously categorized or given Golden Fleece Awards, which are awards established between 1975 and 1987 by that congressperson from
California.
Wisconsin.
Oh, Wisconsin.
Wisconsin.
That's right.
Jeremy Rifkin.
That's,
Is that
No.
him?
No,
No.
he was a gadfly, not a congressman. He
Oh,
was,
sorry.
P begins with P.
Anyway,
Proxmire? No.
it wasn't John
yeah,
Dingle?
I think it was Proxmire. Maybe. Yeah, anyway, these Golden Fleece Awards that were given for things that sounded like really crazy, we're wasting our money on this science stuff. And then explanations of how those turned out to be really important
Proxmire,
research things.
Proxmire.
Yeah, so it was Proxmire. Yeah. Okay. So
very much.
anyway, there's a couple other examples given, and basically she points out that Obama has staunchly supported both curiosity-driven and mission-oriented research, and she suggests that for those of you who want to share similar examples, you can do that using the hashtag basic research. The other one, was how game theory led to had its roots in an analysis of gambling behaviors in 1713, and then that allowed well-designed auctions of the nation's telecommunications spectrum. So just various things that sound really out there, but in the end turn out to be quite practical for the human race.
I would love to add something here. What I find is highly amusing is to hear an announcement. For instance, we're going to conduct war on cancer. Okay, so from now on, all you have to do is just work on cancer and we'll solve it. We'll throw all the money at it. And the very first thing they decide is that we need to do more basic research.
Yes.
Duh.
Yeah.
Jesus H. Sorry, Caleb.
Okay, that's great. I like that. My pick is an article by the man who is laughing because it's by him. It's by Alan Dove. It's called Careers in Virology, Science Writing, and Journalism. And the abstract is very short.
Yes.
This article condenses some highlights from a presentation I've now given at several universities about the bench to newsroom career path for readers who simply want a short explanation of how to parlay their hard-earned critical thinking skills from graduate school into a lucrative job in a growing industry. Go to law school.
Or become an arbitrager.
So, this is Alan's summary of how to...
And you can read the whole thing online because it's...
is it open access?
It's open access,
Did you put that on your CV as a citation?
I'm sure it'll count toward my citation index, which I mentioned in the article, by the way.
Excellent.
Science writing, careers in virology, science writing,
Nice.
and journalism.
Nice.
So he goes through his career, and
Nice.
that's cool. So check that out. Give that a TWiV bump. It'll be read more than any other article in that issue. We have 2 listener picks of the week. Pritesh writes, "Hi TWiV team, here's my pick. Amazing video on how killer T cells kill cancer cells." So it is a video. He sent a link to a Huff post of that, but it's a YouTube video.
And it is very cool.
It's cool.
Yes, it is.
It's beautiful. Very nice. And we have another. Pritesh is at your place, Rich. Do you know him?
Yes, indeed. I'm on his. Am I on his committee? I don't know.
I hope you.
I don't.
No.
No, I don't think so because I'm out of here. But yes, I know Pritesh well.
You're out of here as of what? July 1st.
July 1st.
Going
I got 3.5. I got 3.5 more weeks.
Goodness gracious.
We have to do something.
This
here on
big
TWiV.
party for you, I hope.
We're going to have the party at the ASV meeting. The first thing I'm going to do when I retire is go to a meeting.
Excellent.
So, Rich, are you going to come to ASV every year, or?
If I can get my way paid, yes. Okay.
That's my standard for conferences.
Now you're talking.
It's strictly an economic problem.
Now you're talking.
You're supposed to do. We do TWiV. That's supposed to be a
That'll do it.
in perpetuity payment. You know.
That'll do it. That'll do it. As long as we're doing TWiV, I'm there.
I mean,
because that's where I'm going to get my hit of TWiV and that of virology. Yeah, I would like to.
So you're going to continue TWiV, right?
Yeah, till you kick me out. Yeah,
give me a break. What do you think your frequency will be? Like half of the episodes, roughly?
Oh no, I think it'll be more than that. If you look at the schedule,
yeah.
So the first thing that's happening is my wife and I are taking a road trip.
Nice.
Okay,
So what, like
we're
a
going
year?
to jump in the. It'll last at least a couple of months.
Cool.
Nice,
Okay.
nice.
And it's going to go to Boston and Seattle and Oregon and the San Francisco Bay Area and maybe Southern California and Austin. We're going to circumnavigate the country and connect all the kid dots and just no itinerary, just have a good time.
Oh, Rich,
but
that's
for some of those, we're going to be, sitting for at least 2 weeks in Oregon, and I'll do TWiV while I'm in Oregon. I've done it before. And so sometimes I'll be there, sometimes not. But when I'm home, my guess is it'll be pretty regular. I like this. This is good fun.
All right. Good. Keep up your virology,
Yeah, absolutely. Now, it could be that after a year or so, I'll decide that virology really stinks and I don't want to have anything to do with it. And I'm out of there, okay? But I don't know. This is the magic of retirement.
Session.
I get to discover what's really going on
I'm
here.
feeling you're not
So does your term on the study section end also?
I am going to study section, leaving on Sunday. Study section meets Monday and Tuesday, and that's my last panel.
Uh-huh.
Wow.
You throw your last grant in the box.
Yeah. They don't do that anymore.
They don't
Dickson.
do that anymore. They do that digitally
In
now.
your head, you still do it. I know
Yeah.
you do it.
Yeah, that's right.
cool.
Yep. I'm cleaning up the lab right now. It's
Wow.
really weird. Really
Yeah,
weird.
wow.
I'm going through it from one end to another. All right.
Any
and
surprises?
Throwing out stuff. And I've archived all my viruses. And I went through all the viruses and threw out most of them and made an archive of about 300 viruses that I'm sending to investigators for safekeeping. And everything else went in the autoclave. And now I'm going through the laboratory from one end to another. Do you know how many memories are there?
Oh, tell me about that.
It's
one. I had
amazing.
one like that
It's
too.
just and the
Sure.
Jeez, you got
Data
me going
books.
now. What it reminds me of is why I did this.
Yeah.
Okay, I just like being in the laboratory doing experiments. That's what I want to do.
Yeah, well.
Okay? And just going into the laboratory and cleaning up
So, Rich,
gives me this feeling. You know,
you have an R01 still, though, right?
there's probably about $20 left in it.
Oh, okay. All right. So you terminate when it terminates?
It's amazing the way I titrated this. I titrated it right to the bitter end.
How neat.
Yeah.
Cool. All right. One more listener pick from Ramon. Maybe you could find this TED Talk interesting as a listener pick of the week. It is Romina Libster on the power of herd immunity.
I started looking at it. It's good.
How do vaccines prevent
Oh, yes, I looked at the whole thing. It's quite good.
Immunity should be heard and not seen?
It's in Spanish with subtitles.
Yep.
Neat. And
Very well
Ramon
done.
is our MD friend from Spain. And that's TWiV 340. You can find it at twiv.tv and iTunes and your favorite podcatcher, whether it be iOS or Android. They're all there. Every episode, all free and hopefully there forever. And please do send us your questions and comments to twiv at twiv.tv. Dickson Despommier is right here at Columbia University. Thank you, Dickson.
You're welcome, Vincent.
You can also find him at, medicalecology.com. That would be relevant to this podcast.
Yeah.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
I should remember what you might die of, but I forgot already.
Some kind of atherosclerosis.
That's right.
Yeah, that's it. Chicken wings.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure thing. Always a great time.
When you retire, what do I just say, Gainesville?
Oh, I'll be an emeritus professor.
Oh, you will? That's
Nice.
good.
Yeah.
Are they
Nice.
going to give you an office?
I have been offered an office, but I don't want one.
Really?
I'm gonna, heck, my home is 3 miles from here, and I can do anything from there that I do here. I'll visit now and then, go to seminars and stuff, but it's pointless. It's stupid for me to keep an office. I'm going to spend most of my time doing something else. I've done this for 40 years or more, something like that. I'm ready for
I hear you.
something else.
I hear you.
You have good internet at home, right?
Oh, yeah.
For TWiV, you've done TWiV at home before?
Oh, yeah. Well, if I've done TWiV from home, if it turns out that there's a problem, then I'll come back and get an office at work.
Okay.
I'll find a way. It'll be all right.
Alan Dove is at turbidplaque.com on Twitter. He's Alan Dove. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.